Shoubridge. I'm the chair of the Department of Human Genetics at McGill. My own research lab, which is located here at the Montreal Neurological, works on rare metabolic disorders. We've been doing that for about 25 years. So we try and discover the genes that are defective in people with those disorders, figure out what their protein products do, and hopefully do something about them. The hopefully do something about them is the harder part of that.
And touching on that, I mean, people have swabbed the inside of my mouth, for all kinds
23andMe, did you do that?
And it was a variation of that.
Yeah.
And I find that we're now taking umbilical cord blood, and we're not harvesting it, but we're storing it.
Yeah.
Tell us a little bit about the ubiquity of human genetics today.
So genetics, I mean, because of the tools we have, genetics has taken on more and more and more importance in medicine, in all aspects of medicine, not only in rare disease, but in common disease. Because now we could sequence your whole genome, we could sequence how your genome is modified, and if you have a particular condition, we can look at those sequences and say, well, maybe this is why you have that particular condition, and for you as an individual, maybe there's something we can do about it. But we do it in a kind of personalized way, if you will, based on the genetic information that we gather from you. And that we can do for anybody. We can also edit genes in our bodies. So your example of cord blood was an interesting one, because there you might use the cord blood to do a bone marrow transplant later on in life if you have some problem with bone marrow, stem cells. But we can also go in with new genetic tools that were actually discovered from very, very basic research in bacteria. So bacteria turns out to have an immune system, which is unusual. And the immune system, the things that attack bacteria are called bacteriophages. So they're like viruses that attack bacteria. Viruses attack us the same way. And bacteria have developed a system to recognize those things and chew up their DNA, essentially. And people have, from that very, very basic research, have used those tools from bacteria to allow us to edit the human genome. So we can now go in and change your genome.
That's an amazing spectrum of research and information that we can gather from the information. Tell us about the why behind this research. Why should people get excited about the research that we're doing here?
I mean, I think it's because we're in the midst of a genetic revolution. We've never ever been able to understand the human genome like we can today, and understanding our basic genetic makeup is a key to treating diseases, common diseases and rare diseases, because there's a big influence of our genetic background on how we manifest disease. And we really have to understand that, and that's really I think key, that sort of basic research is key to understanding human disease. And if you don't understand the disease, you don't understand the mechanisms, you can't treat it. So, you can treat a lot of medicine as well. We saw a pattern, and so we diagnosed a disease X. We didn't really know what the genetic basis for it was, or what the predisposing factors might be. But, a good clinician, if they've seen it 100 times, they say, well, it's probably that, because I've seen it 100 times. They say, well, how does it work? I say, well, we don't really know. But we know that this is it, and we know if we give this drug, it makes it a little bit better and may not cure it. So I think the power of genetics is going to allow us to understand, to really get at the basic mechanisms, to really understand disease, and perhaps have drugs that are more specifically targeted without, with fewer side effects, things like that. So I think that's crucial.
Can't wait to follow the progress. Thank you for your time. Are we
Sure.
going to swab the inside of my mouth today?
We could.